So here we are again. We're going to keep trucking along on this train of glucose metabolism. We're in the second half of chapter 19, which is also the second half of the citric acid cycle. And we're going to start this lecture chunk, the first of 3 chunks associated with lecture 14, by first talking about what we've done so far. Let's get ourselves on the same page and discuss the citric acid cycle's first half so that we understand where we're heading into as we go over the second half of the citric acid cycle. That'll be it for this chunk. We'll complete the citric acid cycle, bring ourselves all the way around to oxaloacetate, and then the second chunk will be to discuss and describe how the regulation of the citric acid cycle is achieved by the cell. And the third chunk will broaden our view a little bit and discuss the role of the citric acid cycle in metabolism overall, not only in breaking materials down for energy, but also in making biomolecules using intermediates of the citric acid cycle as precursors for larger molecules that we need to survive. And of course, the side of metabolism that deals with making molecules is anabolism. So, starting where we started the last lecture series, we ended glycolysis with pyruvate, and pyruvate entered the matrix of the mitochondria through a transporter and was immediately acted on by the PDC. The PDC decarboxylated pyruvate, turning it from a 3-carbon molecule into a 2-carbon molecule, and then some subsequent redox reactions converted that into acetyl. Acetyl was linked to CoA as an activation step to make that acetyl a little bit more reactive, and Acetyl-CoA is the molecule that enters the citric acid cycle. The first step of the citric acid cycle was a condensation reaction where acetyl was transferred to oxaloacetate. That made 6-carbon citrate. Once that citrate was made, it was isomerized in step 2 of the citric acid cycle to give us isocitrate. And then we started the first of 2 oxidative decarboxylation reactions, where isocitrate was converted into alpha-ketoglutarate. Carbon dioxide is released. We lost a carbon. That's the decarboxylation. And we also stole some electrons along the way and transferred those electrons to NAD, creating NADH plus H. Once we made alpha-ketoglutarate, we did another oxidative decarboxylation, lost another carbon as carbon dioxide, and converted alpha-ketoglutarate into Succinyl-CoA, again stealing electrons and protons along the way. And that's where we left off. We left off with Succinyl-CoA in the middle of the citric acid cycle, and in total, if we also include the PDC, we've reduced 3 molecules of NAD plus to NADH plus H, stealing a total of 6 electrons. And we also released 3 molecules of carbon through carbon dioxide release. Keeping in mind here that we're doing this in parallel 2 times over for every molecule of glucose, because for every molecule of glucose we made 2 pyruvates, converted into 2 acetyl-CoAs, run through 2 citric acid cycles. We've just lost the equivalent of 6 carbons through these 2 parallel cycles, and that's the entirety of our glucose. So the carbons from our glucose molecule are now officially gone. The rest of the citric acid cycle is going to be to do just that, to make it a cycle, to recycle back to oxaloacetate. So let's pick up where we left off at Succinyl-CoA. Like any CoA bond, the bond between succinate and CoA is a high-energy bond. It's very unstable. Succinate is a stable, non-reactive molecule, and it's made reactive through this CoA bond. And so we can easily break that bond, separating succinate and CoA, and we do that here through a hydrolysis reaction. Because that energy bond is so high energy between succinate and CoA, a lot of energy is released in this hydrolysis reaction, and we capture some of that energy by phosphorylating GDP into GTP. So we actually make a triphosphate here. The enzyme that catalyzes this reaction is called Succinyl-CoA synthetase. I'll say that again, synthetase. Of course, this enzyme is named for the opposite reaction. We're breaking Succinyl-CoA down, we're not making it. But more importantly, there's something to keep in mind here about enzyme names for synthetic enzymes, something that's going to help us out as we continue down on this road of glucose metabolism. Both enzyme classes called synthases and synthetases are responsible for anabolic reactions, reactions that make new bonds and therefore make new biomolecules. So embedded in each of these enzyme class names is synthesis, so we're making things. So why have 2 different enzyme names for 2 classes of enzymes that do the same thing, that make things? Well, there's a subtle difference between the 2. Synthases make new biomolecules but they do so using energy derived from something other than ATP. On the opposite side, synthetases also make new biomolecules, but the energy they need comes from ATP, either directly or indirectly. Most of the molecules that our cells make require ATP for synthesis, so most of our synthetic enzymes are synthetases. But there are a few, and we'll encounter them later on in this course. There are a few enzymes that make things without needing the energy from ATP, and so those are our synthases. So again, if we're naming this enzyme for its opposite reaction, essentially this enzyme name tells us that the enzyme can make Succinyl-CoA using energy from ATP, directly or indirectly. So that stands to reason then that when this enzyme engages in the reverse reaction, it breaks Succinyl-CoA down and can make ATP. In other words, restore the energy that would have been used had Succinyl-CoA been made. The energy to make that ATP comes from the hydrolysis of succinate and CoA. We already said that. But I told you that this enzyme makes GTP, not ATP. So let's follow exactly what's going on here. This is a crystal diagram, an X-ray crystal structure of Succinyl-CoA synthetase. And living right here in the active site, we see a GDP molecule. That's going to be the GDP that accepts that phosphate and is converted to GTP. So we're making GTP. That means we need energy. Making ATP is endergonic. We already said the energy to make that molecule comes from the hydrolysis of succinate and CoA. That energy released by breaking that unstable CoA bond is roughly equivalent to the enzyme that we need to phosphorylate GDP. So from an energetics perspective, the delta G of this reaction is pretty neutral. It's pretty close to zero. The universe really doesn't care energetically how we do this. So this reaction is governed by the law of mass action. It's a set equilibrium. If there's more reactant, we'll go in one direction. If there's more product, we'll go in the other direction. Once that GTP is made, though, another enzyme comes in, and the name of this enzyme is nucleoside diphosphate kinase. Let's break that enzyme name down. Nucleoside diphosphate kinase. It sounds like this is an enzyme that can take a nucleoside diphosphate, and probably any nucleoside diphosphate, and act on it as a kinase. In other words, phosphorylate it to a triphosphate. And indeed, that's exactly what this enzyme can do. It's a promiscuous enzyme. It can work on almost any nucleoside diphosphate, and it can phosphorylate those into the triphosphates. So here's a side reaction in this step. This enzyme takes that newly made GTP that was just created by Succinyl-CoA synthetase and uses the third phosphate of that GTP. That's the third phosphate this GTP just gained, and takes that phosphate and transfers it to ADP, creating a molecule of ATP. This is also substrate level phosphorylation. We're phosphorylating an ATP molecule using a single donor of a single phosphate at the substrate level. And we can summarize the reaction catalyzed by nucleoside diphosphate kinase in this simple diagram here. We start off with GTP and ADP, and once the enzyme does its job, that last phosphate is transferred to the ATP, giving us guanosine diphosphate and adenosine triphosphate. This is the only point in the citric acid cycle where we will make any type of usable energy. The rest of the citric acid cycle is for harvesting electrons, as we've already said. And it's almost as if making this single ATP molecule is an afterthought. It's almost like, hey, look, all of a sudden we're releasing a bunch of energy. Well, let's go ahead and capture that before it's wasted. And that is the philosophy behind this step. So to summarize it, we're taking Succinyl-CoA, hydrolyzing it to succinate. CoA is recycled back to be linked to either acetyl or another succinate. And we use the energy of that hydrolysis to phosphorylate GDP into GTP, and then we instantly trade that phosphate onto ADP through the action of nucleoside diphosphate. So essentially, from the perspective of the citric acid cycle, we just made a molecule of succinate. As I told you last week, once we get about 2/3 of the way through the cycle, and that's where we are now, the rest of it is for the regeneration of oxaloacetate, and we're at that point now. We're just trying to get back to our primary intermediate, our key intermediate, so that we can cycle, so that we can do this again. There are 3 more steps to the citric acid cycle, steps 6 through 8, and they're summarized very quickly here. We'll go over them in a bit more detail in just a moment, but to show you where we're heading. We're going to oxidize succinate to fumarate and reduce FAD along the way. We're going to hydrate fumarate to malate. And then one last chance here, harvest as many electrons as we can. We will oxidize malate to oxaloacetate, transferring those electrons and protons to NAD. This will essentially convert a 4-carbon molecule succinate into a different 4-carbon molecule oxaloacetate, again harvesting electrons as we do so. So in step 6, this is a redox reaction. Succinate is oxidized to fumarate. We can see where those electrons are coming from. They're coming from these central carbons, carbon 2 and 3 here. We lose protons from those carbons and electrons as well, and those 2 electrons and protons are used to reduce FAD to FADH2. The enzyme that catalyzes step 6 is the only enzyme of the citric acid cycle that you must know by name. Remember, I told you that we're just essentially after the reaction types of all of the other reactions, and step 6 is catalyzed by succinate dehydrogenase. You'll see why in just a moment, why I want you to know this enzyme. It is the most distinct and arguably the most important enzyme of the citric acid cycle. I mentioned to you in the last lecture that all of the reactions of the citric acid cycle occur in the mitochondrial matrix, deep in the mitochondria, floating in the center of the organelle, except for one. This is the step that is not in the mitochondrial matrix. Instead, succinate dehydrogenase is actually an integral protein, and the membrane that it is embedded into is the inner membrane of the mitochondria. Remember, integral proteins are proteins that are embedded in membranes. So succinate dehydrogenase lives within the mitochondrial membrane, the inner membrane of the mitochondria. And so that's where this reaction occurs, on the inner leaf of the inner membrane of the mitochondrial organelle. Also, as you can see, unique for the citric acid cycle, this is the first and only time where we will reduce FAD, not NAD. FAD is not a mobile electron carrier. FAD cannot move. It is bound to the enzyme itself. And so these electrons are kind of sort of stuck on this enzyme, but kind of sort of not of, as you'll see in just a second. So FAD is usually covalently bound to whatever redox enzyme or dehydrogenase enzyme is using it, and that's the case for succinate dehydrogenase as well. FAD is stuck to that enzyme. Last, and most importantly, succinate dehydrogenase is distinct because it does double duty. It's not only part of the citric acid cycle, it is also part of the electron transport chain, which is where we will be going in our next lecture. Very briefly here, this is the electron transport chain in its most simple consideration. The electron transport chain is made up of 4 different protein complexes, and then the last 5th complex is the one that actually makes ATP. They're very succinctly abbreviated as complex 1, complex 2, complex 3, and complex 4. Here we see complex 5 as the complex that makes ATP. But I want to zero in very quickly on this figure legend and see that complex 2 goes by another name, succinate dehydrogenase. So this enzyme that catalyzes step 6 of the citric acid cycle is actually also part of the electron transport chain. It's okay that FAD is stuck to this enzyme, because the electrons are going to be offloaded off of succinate dehydrogenase and fed directly into the citric acid cycle. We'll expand on this idea in lecture 15. In any event, succinate is oxidized to fumarate, with succinate dehydrogenase stealing the electrons and protons and putting them on its connected electron carrier, FAD. Now in step 7, a hydration reaction where we add a single water molecule to fumarate, creating malate. We can see here that fumarate is made into malate when we take this water molecule and split it, and put 1 proton on carbon 3 and the hydroxyl group on carbon 2. That breaks the double bond between those 2 carbons. This reaction is catalyzed by an enzyme named fumarase, which you will not need to know by name. And malate is chiral. If you look, you can see there are chiral carbons there. This carbon 2 is chiral, specifically. And interestingly, only the L form of malate is ever made by fumarase. And this is important because in the last chunk of this lecture series, in chunk C, we're going to see how we can steal from these citric acid cycle intermediates to make amino acids, of all things. And amino acids, of course, are chiral as well. So we're setting the stage here for making L-form amino acids by making L-form malate. And we saw that before with aconitase and isocitrate. So we are there in step 7, fumarate converted into malate through a hydration reaction. And finally, step 8 is the step where we will regenerate oxaloacetate and complete the cycle. In addition to this being the last step of the citric acid cycle, it's also our last dying gasp of stealing electrons from this glucose intermediate, and so this is our last redox reaction of the citric acid cycle. Malate is oxidized, electrons and protons are stolen from it, and it is oxidized to yield oxaloacetate. NAD is what is reduced in this reaction to NADH plus H. And we can see that reaction right here. We see the electrons and protons that are stolen are coming off of carbon 2, and they're being linked onto NAD. This reaction is catalyzed by malate dehydrogenase, a wonderful name for this enzyme, telling you that malate is having hydrogen stolen from it. And of course, where the hydrogens go, the electrons follow. So we've come around to the beginning. Once we've remade oxaloacetate, we've reestablished that key intermediate, that primary intermediate of the citric acid cycle, and we're ready to take another acetyl molecule, link it to that oxaloacetate, and go around the citric acid cycle again. The citric acid cycle is nothing but an electron harvesting series of reactions, and we've come back to the beginning so that we can harvest electrons from another round of acetyl-CoA. To summarize the citric acid cycle energetically, if we look at the citric acid cycle overall, we see that if we include consideration of the PDC, we've released 3 molecules of carbon dioxide through decarboxylation reactions. We've made 1 molecule of ATP through the action of nucleoside diphosphate kinase. 1 molecule of FAD was reduced to FADH2, and 4 molecules of NAD were reduced to NADH plus H. That's a total of 10 electrons that we have ripped off of what was once glucose. 6 of these electrons have come from the citric acid cycle itself, and 2 of those electrons have come from the actions of the PDC. Although we've only made 1 ATP from the citric acid cycle and 2 ATPs from glycolysis, we've gone through quite a bit of biochemistry, and all we have to show for it is 3 measly molecules of ATP from all of this. Remember, we are not done yet. Everything we've done has been done aerobically. This was all an investment. It's the electrons that we're after, it's the electrons that are carrying our energy, and we will reap large rewards for all of this time and effort as we continue to the end of aerobic respiration, where we will make ATP by the dozen. The next question becomes, if the citric acid cycle is so important and so critical, if it's so dedicated as well, if it's this electron harvesting mechanism, how is it controlled? How do we regulate the citric acid cycle? And that's what we'll begin with in the next lecture chunk. So I'd like to leave you with the roadmap here. We will not be returning to this again. Be sure that you understand the general purpose of the citric acid cycle, have a very strong sense of what's occurring in each step from a reaction type point of view, and don't lose track of our 2 key points from this. 1, the idea of an oxidative decarboxylation reaction in general, and 2, succinate dehydrogenase and all that's involved in that step and with that enzyme. I will see you guys in chunk B whenever you choose to watch it.